with telebastat, the DPP9 inhibitor, we see an increase in IL-1 beta levels, which is then reduced when we have knockout of NLRP1, caspase-1, and ASC, and is unaffected by knockout of caspase-11 or NLRP3, and these are two independent inflammasome sensing proteins. As a control here, we've used nigericin, which is a bona fide NLRP3 activator. These results were also held consistent in human cells. So again, we have human immortalized keratinocytes that were treated with telebastat, which induces an IL-1 beta response, which is nearly completely lost when we have knockout of NLRP1 or the adaptive protein ASC. So when we inhibit DPP9, we get NLRP1 activation. So the conclusion from this and several other papers was that DPP9 was working to inhibit NLRP1 inflammasome formation. As its name would suggest, DPP9 is a peptidase that cleaves the last two amino acids of the N-terminus of proteins, where there's a proline here in the second position. DPP9 has a wide variety of roles in the cell, including immune activation, apoptosis, and cell migration. So a lot of work was done to determine exactly how DPP9 was inhibiting NLRP1, but it was really clarified in these two papers here that were published back-to-back in Nature last year. They both solved the structure of DPP9 bound to NLRP1. One of these papers solved the structure of the human proteins, one solved the structure of the rat proteins, and found essentially the same thing, that one molecule of DPP9 was actually bound to two molecules of NLRP1, where one of these molecules was the full-length protein and the other was just the C-terminal fragment. So quite interestingly, I think, despite NLRP1 having this proline cleavage motif, DPP9 was not found to cleave NLRP1, and this has been confirmed by several different studies now. So to put this all together in a schematic, at baseline, DPP9 restrains NLRP1 inflammasome activation by binding the inflammasome-forming C-terminal protein. And in doing so, it acts as kind of like a sponge to stop it from oligomerizing and forming an inflammasome complex. But when DPP9 activity or binding to NLRP1 is reduced, the C-terminal fragment is then free to oligomerize and form this inflammasome. So going back to the patients with the NLRP1-driven autoinflammatory disease, in a follow-up study, Zhong et al. Again expressed a variety of different patient mutations in cells and then did an immunoprecipitation of NLRP1 to look for binding of DPP9. And interestingly was this variant, which was a P1214R substitution, and this variant caused DPP9 to no longer bind to NLRP1. And perhaps it is this lack of DPP9 binding that is leading to the NLRP1 overactivation in this patient. So that was our first kind of hint that DPP9 restraint of NLRP1 was physiologically meaningful. And before this, all the work looking at DPP9 inhibition of NLRP1 has been done in vitro using cell lines. So for my PhD project, we wanted to look further in the in vivo consequences of DPP9 regulation of NLRP1. So I'd really like to highlight that this project has been, I think, a really nice collaboration between 3 different labs. The Reverse SARD and the Zhong lab at ASTA in Singapore did a lot of work around human patients, and then my part in the Masters lab was to do in vivo mouse work. We currently have a preprint up on medRxiv if you are interested in reading about this. So the Reverse SARD lab, Bruno and his postdoc Kenneth, have identified 2 patients from 2 separate families that both have loss-of-function mutations in DPP9, and these patients display an autoinflammatory disease. The patient from the first family has a compound heterozygous mutation, where one of these mutations results in the introduction of a premature stop codon, and the other is an amino acid substitution at position 167, and this is a highly conserved region of DPP9. The patient from the second family has a homozygous mutation, where again this results in the introduction of a premature stop codon. So the Zong lab, Franklin and his postdoc Kim, took primary fibroblasts from these patients and looked at DPP9 expression via Western blot. So first looking in this fifth lane here, we have the cells derived from the patient with the homozygous nonsense mutation, where we do not see any evidence of DPP9 expression. In the other patient with the compound heterozygous mutation, we see some DPP9 expression, however, it is reduced compared to wild-type cells, and this is consistent with one of these alleles being a premature stop codon. The Zong lab wanted to explore that whether this missense mutation in this patient changed the activity of DPP9. So they performed an enzymatic assay looking at the ability of different DPP9 variants to cleave a canonical substrate, and did in fact see a significant reduction in the enzymatic activity of this patient variant compared to wild type. So overall, these patients lack either DPP9 expression or catalytic function. In terms of symptoms, both the patients show skin phenotypes, much like the patients that have the NLRP1-driven autoinflammatory disease. They both had a general failure to thrive, had learning difficulties, including speech delay, as well they were of proportionate short stature. In terms of immune phenotypes, they suffered from repeated infections, had pancytopenia, and also suffered from bronchitis. So a Western blot of primary keratinocytes taken from the patient with the homozygous nonsense mutation showed that there was inflammasome activation in these cells. So to take you through this Western blot, we can see that there was cleavage of IL-1 beta and IL-18, which I'll remind you are the two cytokines cleaved by the inflammasome complex, as well as evidence of oligomerization of ASC, which again is evident of this formation, this complex being formed. Furthermore, these patient cells showed an increase in IL-1 beta and IL-18 levels seen in an ELISA, and none of this evidence of inflammasome activation was present in cells that were taken from this patient's parents. So overall indicating that the inflammasome is indeed active in these patient cells. So we wanted to further explore this disease, and to do so, we utilised a mouse model that had a mutation in DPP9 that renders the protein catalytically inactive. So these mice were made and characterised by the Gorell group, which is a group at the Centenary Institute. And here is just another enzymatic assay showing that mice, or cells from these mice that are homozygous with the DPP9 mutation, lack catalytic activity compared to wild-type cells. So at birth, mice that were homozygous, heterozygous, and wild-type for the DPP9 mutation were all present at the expected Mendelian ratios. However, quite interestingly, all DPP9 homozygous mutant mice died within 24 hours of birth and hence showed a complete early neonatal lethality. So this group went on to characterize these mice and, despite lots of work, could not find an obvious reason for death. Here I'm showing you some histology they performed, comparing major organs between DPP9 wild type and homozygous mutant mice, where no difference was apparent. So we know that DPP9 restrains NLRP1 inflammasome activation. So we were wondering that if the lack of DPP9 activity in these homozygous mutant mice was causing overactivation of the NLRP1 inflammasome, and could this be the cause of the neonatal lethality? So this led us to the hypothesis for my PhD project, which was that knocking out NLRP1 and downstream inflammasome components would rescue the death seen in the homozygous, the DPP9 homozygous mutant mice. So today I'll be discussing two aims regarding this hypothesis, the first of which is to determine if and how NLRP1 overactivation leads to death in the mice. So to do this, we crossed mice that were knockouts for NLRP1 to the DPP9 mutant mice, and really excitingly, we did see rescue of the neonatal lethality. So in this table, I am showing you the number of mice that were present at weaning, which is roughly 3 weeks of age, as a result of this cross, where I have the observed number of mice here on the left, with the expected number in brackets. So, as we would predict, there were no DPP9 homozygous mutant mice that were wild type for NLRP1 surviving to weaning. However, we did see survival of mice that had a heterozygous or homozygous loss of NLRP1, indicating that rescue has occurred. This rescue had an incomplete penetrance, where we saw about 12% of the expected NLRP1 heterozygous mice surviving, which went up to about 26% for the NLRP1 homozygous mice. So, having established that NLRP1 is clearly playing an important role in the neonatal lethality, we wanted to look at exactly how it was doing this. So, I collected mice within a few hours of birth, and as the group that made the mice saw, we again saw the expected Mendelian ratios of all the different phenotypes at this early time point. I also took images of all the pups, and we can see that the DPP9 homozygous mutant mouse, wild type for NLRP1, appears normal and appears the same as the DPP9 heterozygous and wild type glutamate controls. Furthermore, these mice showed evidence of a milk spot, which is indicative that they are indeed suckling and feeding. So, as the DPP9 deficient patients displayed pancytopenia, we wanted to determine if there were defects in the hemopoietic system that could be contributing to death. So, I analysed the blood of these newborn mice and looked at various different immune cell subtypes, including T cells, B cells, monocytes, neutrophils, NK cells, and dendritic cells. And when looking at DPP9 homozygous mutant mice, again wild type for NLRP1 knockout, we saw no difference in survival compared to the DPP9 heterozygous or wild type controls. I also looked in the bone marrow of these same mice, and again, we saw no difference in any of the immune cell subtypes I measured. I also wanted to look at the ability of the bone marrow progenitors to differentiate. So again, I took bone marrow from these mice. I differentiated them with either MCSF or GMCSF. At the end of about a week of differentiation, the DPP9 homozygous mutant bone marrow looked normal, and when looking at the differentiated immune cell subtypes, we again see no difference between any of the genotypes. Finally, I also collected the liver from these newborn mice, and again, I saw no difference in any of the cell subtypes measured between the DPP9 homozygous mutant and wild-type pups. So overall, I couldn't identify any defects in the hemopoietic system of these mice, likely showing that this wasn't contributing to the neonatal lethality. So that's as far as I got in my analysis of the DPP9 homozygous mutant mice at birth, but we had these rescued DPP9 homozygous mutant NLRP1 knockout mice, and we were really interested in studying them because by doing so, we can be able to maybe determine what NLRP1 dependent and independent effects of DPP9 loss of function, and perhaps in doing so, this would tell us a bit about the patients. So I aged a subset of these mice to look at their lifespan. As a high proportion of the DPP9 homozygous mutant NLRP1 knockout mice did die before weaning. I only included mice that were alive at the time of weaning in this cohort, indicated by this dotted line here. So, to really drive the point home, all of the DPP9 homozygous mutant mice that are wild type for NLRP1 die within 24 hours. However, our rescued mice in the red line show a comparable lifespan to our control mice, which here are the DPP9 heterozygous NLRP1 knockout mice. There has been no identified effect of DPP9 heterozygosity, and these mice appear perfectly normal, which is why they're our controls. There was a slight, decrease or trend to a decrease in the survival. However, this was not statistically significant. Okay. So the first phenotype we notice about our rescued mice is that they were runted compared to the controls. So I think it's best or easiest to show you this runting in a video I took of these two mice at 2 to 3 weeks of age. So these are two litter mates. They're both males, and I think it's very obvious the size difference in the DPP9 homozygous mutant NLRP1 knockout compared to the control mouse. A few weeks later, at about 5 to 6 weeks, again, these are the exact same mice. The size difference is still apparent. However, I do think this mouse is looking a lot more healthier than it was previously. So to quantify this runting, we weighed the mice weekly for about 9 weeks, starting at weaning. And perhaps unsurprisingly, given those videos, we see a significant reduction in weight in the DPP9 homozygous mutant mice that have either a heterozygous or homozygous loss for NLRP1. And this runting we can see this runting throughout the entirety of the mouse's lifespan, where again, here I'm showing you two male litter mates. This time they are 6 months old, where the size difference of our rescued mouse is a lot smaller than the heterozygous control. So we wanted to explore exactly what was contributing to the runting we see in these mice. And the first way in which we did this is with the help of Lucky. I scanned some mice using the micro-CT, and in doing this, this allows us to make a 3D reconstruction of the skeleton, where I can then measure the precise lengths of bones to determine if there are any skeletal differences that are contributing to the runting. So, as I've used both males and females in this analysis, I've plotted the bone lengths as a percentage of the same-sex control mouse in order to remove any variability due to sex. But when I do this, we see a decrease in hip length and hip width in the DPP9 homozygous mutant NLRP1 knockout mice. Their lengths of their limb bones remain reasonably consistent, perhaps with the exception of the humerus, which also reached a significant decrease, overall indicating that yes, there are skeletal differences contributing to the runting. I also wanted to look at organ and tissue weights. So to do this, I collected mice at 6 months of age, and again, unsurprisingly, we see a significant reduction in the weight of our runted mice, with a trending but not yet significant reduction in the carcass weight, where here carcass is referring to the skeletal and muscular system. Quite interestingly, we saw a reduction in the runted DPP9 homozygous mutant NLRP1 knockouts in their subcutaneous and visceral fat, where subcutaneous fat is the fat that's under your skin and visceral fat is the fat that's around your organs. So as these mice also had a lower total body weight, I've also plotted these weights as a percentage of whole body weight. And in doing this, we still see a significant reduction in subcutaneous and visceral fat of these mice. And this is indicative that they have a reduced body fat percentage. As for the internal organs of these mice, when plotting the raw weights, the DPP9 homozygous mutant NLRP1 knockouts have a reduced weights of the digestive tract as well as the liver. But when I plot this as a percentage of total body weight, this reduction is no longer seen. So overall, the runting we see in these mice appears to have be the cause of both skeletal differences as well as a reduction in body fat percentage. We histologically examined these mice, and one of the only phenotypes we found is that they had immune cell infiltrate in their lungs. So here I'm showing you 6-month sections from 6-month-old mice where we can see the immune cell infiltrate is kind of localizing around the bronchioles in the DPP9 homozygous mutant NLRP1 knockout mice, which is not apparent in either the heterozygous or wild-type controls. Immunohistochemical staining of these sections revealed that we had F480 positive cells, which is indicative of macrophages. I don't know how well you can see them, but these macrophages were quite large and looked almost bubbly. So to me, I think they look like foamy macrophages. We also had CD3 positive and B220 positive staining cells, indicative of T cells and B cells in the immune cell infiltrate. So as I mentioned, these were lungs taken from 6-month-old mice. I also took lungs from our age cohort. So these are mice that were aged to well past the year, and the immune cell infiltrate we see appears to be a lot more severe at this time point, and this may explain the slight decrease in survival we see of this in this genotype of mice. So, as I previously mentioned, given that the DPP9 deficient patients experience pancytopenia, we wanted to look in the blood of the rescued mice. So to do this, we did an ADVIA analysis, and I won't show you every single parameter today, but overall, there were no differences in any of the parameters measured in the DPP9 homozygous mutant NLRP1 knockout mice. So at this point, I'd like to draw some comparisons between the DPP9 deficient patients as well as the DPP9 homozygous mutant mice, both with and without NLRP1. And in doing this, we can begin to tease apart what are some NLRP1 dependent and independent consequences of DPP9 deficiencies. So I'll begin with the similarities between the patients and our rescued mice. So the patients show a failure to thrive and are of short stature. This is mirrored in our mice in that they are runted. The patients experience bronchitis. This is mirrored in our mice in their lung cell immune infiltrate. As these mice are knockout for NLRP1, we believe that these presentations in the patient are NLRP1 independent consequences of DPP9 loss of function. But clearly there are some NLRP1 dependent consequences. So I put failure to thrive here, again, because all the DPP9 homozygous mutant mice die within 24 hours of birth, but once you knock out NLRP1, some are rescued, indicating that NLRP1 is playing a role in disease. There were also a few presentations in the patients that we could not attribute to being NLRP1 dependent or independent. The patients have learning difficulties and had repeated infections. We did not measure these in the mice. The patients had skin lesions as well as pancytopenia. Both of these things were absent in both the DPP9 homozygous mutant pups and our rescued mice, and this could be just due to the fact that humans and mice are quite different. So at this point, I'd like to summarize my first aim. So I've shown that the neonatal lethality caused by DPP9 homozygous loss of function can be rescued when we knock out NLRP1. After weaning, these rescued mice show a comparable long-term survival to control mice. However, they are runted and they display a general immune cell infiltrate in their lungs, but otherwise they appear healthy, and both males and females are fertile. So moving on to my second aim, we wanted to cross the DPP9 mutant mice to mice that had deletion of other components of the inflammasome pathway. And in doing this, we could really determine or confirm that the rescue we saw with the NLRP1 knockout is specifically due to the overactivation of the inflammasome. So firstly, we crossed our DPP9 mutant mice to mice that were knockouts of gasdermin D, which I'll remind you is this pore-forming protein that allows for cytokine release as well as cell death. So we also saw rescue when we knocked out gasdermin D. In this graph, I'm showing you the penetrance of rescue, where I plotted the number of DPP9 homozygous mutant mice that are alive as a percentage of what we would expect via Mendelian ratios. So much like the NLRP1 cross, both homozygous and heterozygous knockout of gasdermin D also rescued the mice, and again, this rescue was incompletely penetrant. So I analysed these rescued gasdermin D knockout mice in much the same way I did the NLRP1 cross, and I won't dwell on this data, I'll quickly go through it. But essentially, our gasdermin D rescued mice showed comparable survival compared to the DPP9 heterozygous controls. However, I did only take this analysis up to 1 year of age. The DPP9 homozygous mutant gasdermin D deficient mice also had reduced weights compared to the DPP9 heterozygous controls, indicating that they were runted. Micro-CT analysis again showed this runting was at least partially due to differences in the skeletal system. Again, we saw immune cell infiltrate in our DPP9 homozygous mutant mice with knockout of gasdermin D. And again, this lung cell immune infiltrate stayed positive for F480, CD3, as well as B220. And finally, in an ADVIA analysis, again, I'm not showing you all the parameters because there was no difference we saw when comparing the DPP9 homozygous mutant, this time a heterozygous knockout of gasdermin D, compared to the DPP9 heterozygous controls. Overall showing that these rescued gasdermin D mice phenocopy that of their NLRP1 counterparts. So we also crossed the DPP9 mutant mice to mice that were knockouts of the adapted protein ASC, as well as IL-18 knockouts and IL-1 receptor knockouts, where IL-1 receptor is the receptor for IL-1 beta. And we did this because we wanted to tease apart their individual contributions to disease. So again, I'm showing you a very similar graph to before, where I'm showing you the penetrance of rescue for each of the crosses. We also saw rescue when we knocked out the IL-1 receptor, as well as the inflammasome adapter protein ASC, and we did not see any rescue with the IL-18 knockouts. And consistently with our previous cross data, all of these rescues showed incomplete penetrance. And initially, we thought this incomplete penetrance was because the mice were runted, and we thought that the rescued mice were being cannibalized or perhaps neglected by their mothers because of this runting. So to formally address this, we also collected mice at 2 days of age to genotype them, and we chose this time point because at this time point the mice are not runted, at least visibly. And really interestingly, we again only see this incomplete penetrance of rescue with all the genotypes we tested, so the NLRP1 knockouts, the gasdermin D knockouts, and the IL-1 receptor knockouts. So I think this is quite interesting because clearly NLRP1 inflammasome activation is contributing to the death in these mice, as we can rescue them, but DPP9 has a lot of functions outside NLRP1 regulation. So perhaps these other consequences of DPP9 loss of function is also contributing to the lethality. And by knocking out the NLRP1 inflammasome, we can kind of tip the scales in enough of these mice that some of them survive, but some don't. The last thing I really wanted to talk about is that we saw rescue with the IL-1 receptor knockout, but not the IL-18 knockout. And this indicates that IL-1 is playing a larger role in disease pathogenesis as compared to IL-18. And this is really interesting to know, as there are anti-IL-1 therapies that have proved beneficial in autoinflammatory disease patients. And to back up this cross data, again, I'm going to go back to the NLRP1-driven autoinflammatory disease patient that had the P1214R mutation that lacked DPP9 binding, because this patient was given an IL-1 receptor antagonist, and this proved beneficial for them. So this, combined with our cross data, suggests that anti-IL-1 therapies may be really beneficial in our DPP9 deficient patients. So in summary of my second aim, I found that the neonatal lethality caused by DPP9 loss of function can also be rescued by knocking out the inflammasome adaptive protein ASC, the pore-forming protein gasdermin D, as well as a receptor for IL-1, but not IL-18. Furthermore, the rescued DPP9 homozygous mutant gasdermin D knockout mice phenocopy that of their NLRP1 counterparts, and the incomplete penetrance we see at weaning is also observable at 2 days of age, which is indicative that there are other functions or other consequences of DPP9 loss of function that are contributing to the early neonatal lethality. So ultimately today, I hope I've shown you that we and our collaborators have identified a new autoinflammatory disease that's caused by deficiency of DPP9, and this leads to the activation of the inflammasome. And by modelling this disease in mice, we showed an important role for NLRP1 in driving disease, and perhaps by dampening NLRP1 signalling with an anti-IL-1 therapy, this might be beneficial in our DPP9 deficient patients. Okay, so I'm running a little bit early, so I'm going to really draw out these acknowledgements. I'd very, very much like to thank our collaborators, Bruno, Franklin, and their postdocs, Kim and Kenneth. None of the human data would have been possible without their work. I would also very much like to thank everyone else that has contributed to the paper and really acknowledge the patients and their families. So I'd really like to thank my supervisors, Seth and Alan. I'm going to have some water. I really appreciate all that you've done for me over the past few years. Seth, thank you for hiring me as an RA. I think that turned out well. I also appreciate the independence you've given me throughout my PhD, as I think that's really helped me develop as a scientist. Alan, thank you for all your advice and mentorship and support. It's been really, really valuable, and I'm really proud of you for now having your own lab. It's going to be great. Thank you to everyone else in the Masters Lab, both past and present members. I've had a really good time in this lab, and thank you to each one of you for making it a really happy, positive environment to work in. Specifically, thank you, PowerApp, for being my NLRP1 buddy, who I can talk for hours about NLRP1. Thank you, Clara, for doing some bioplex experiments that I did not show today. And thank you, Sophie, for being a really great support and mentor. I'd like to thank my committee, Sandra, Danny and Ruth, for the advice they've given me over the last few years. I'd like to also thank the inflammation division and these people in particular. I won't go through individually, but they've all provided me with reagents or protocols and whatnot, which have been helpful. In particular, thanks to Jane and Tobias for keeping our division running. I'd really like to thank Bioservices. My project is entirely mouse-based, as you saw, and I could not have done the work I did without them. So in particular, thank you to Sophia, Eden, Tom, and the entirety of Room 12. I'd like to thank Lockie from Imaging for help with the micro-CT, as well as the histology and fax lab. I would like to thank my parents and my family for all their support. My parents used to work in science, so they know all the ups and downs. And, always give me lots of advice, which I may or may not listen to. Finally, thank you to Daniel, who has been a source of never-ending support. And thank you to our cats, Peanut and Pumpkin.
That's fantastic, Cass. Thank you so much for a wonderful presentation. I know there was a problem with Slido, and as we have time, if I might just restate a couple of things Alan said at the start, because, as your other supervisor, I've had been so impressed with your output over the last few years. It's been really amazing, and these are really hard experiments. I'm sure you saw, the analysis of mice at 1 or 2 days of age is really tough, and so Cass really went above and beyond to gather the data that you saw. And it's really important. I can't, perhaps it just seems like, well, there's a couple of people with this disease, but to be able to rescue the mice tells us about how to treat all those patients in the future. And I got an email this morning, there is another DPP9 deficient patient out there, and that's going to be true forevermore because genetic abnormalities always happen at different points in time. And so this sort of information is going to be important in the future. The thing about Cass, that distinguishes her as a scientist, if I can just steal 1 more minute of question time, is her ability, the true quality of a scientist is to be excited by the data, and she is constantly, and that's fantastic. But then when that data turns out to be incorrect, you can't be totally down in the dumps. You've just got to dust yourself off, get up and get the next piece of data, and that she does as well. And so I've been really impressed with your qualities as a scientist, Cass. Now, I will let you ask all of those questions you're burning to ask. For those of you in the audience and in Slido, do we have a few hands? Anna, I know straight away.
So I've got a couple, if I may. You had your table where you were comparing the phenotype of the symptoms of the patients to the phenotype of the double knockout mice. And I was sort of a little bit curious about the concept, because the patients are, of course, NLRP1 wild type, presumably. So you wouldn't actually expect most of the inflammatory symptoms. You would still expect these deficiencies to the other, based on the other roles of DPP9, which they probably aren't clear. But there you have the problem that you're working with inbred mice, and the DPP9, once you've on an outbred background, might actually live. I don't know if you've ever considered that. So that's sort of one thing. The fact that they don't have skin lesions, for example, the mice, well, maybe they need NLRP1 to get those skin lesions. So it didn't really, that comparison was a bit curious.
Yeah, so I think I put that comparison up there to really show that there are NLRP1, I guess, dependent consequences of DPP9 loss of function, but there are also clearly other things that are happening that are NLRP1 independent, and with the knockout mice that we've rescued, we can kind of use them to tease that apart. Also, as those knockout mice do, survive till adulthood, we can, well, maybe not we, but other people may be able to use them as a tool to look at other DPP9 functions, as they no longer have to deal with, mice that are dying right after birth.
Yeah.
Yeah. But yes,
But the major problem, well, not the major problem, I suppose, but one of the debilitating problems of the patients is the inflammation.
Mm-hmm.
And so that's not fully represented once you delete NILP.
Exactly.
So the other question I had, so where you have just the DPP9 mutant, homozygous mutant, and they all die within 24 hours of birth, has anybody looked if that is a septic process? As soon as the gut is populated by bacteria, have you tried or have you considered raising them germ-free, for example, or essentially protecting them in some other way at that stage?
Yes, so we do think it's clearly something is happening either at birth or within those first few hours that could be causing the activation of NLRP1. And as DPP9 is no longer there to restrain it, it goes crazy. We haven't done any germ-free experiments, nor have any other labs. But yes, we do think microbiome colonization is a pretty, I don't know, it's definitely possible, and that's what's causing the death, but we haven't formally tested it.
But I think that's a good question, and Jacob Jackson thinks that's a good question as well on Slido. So,
Hi, Kath. Great talk. Congratulations on great work. I really enjoyed your talk.
Thank you.
So my question is, loss of NLRP1 doesn't offer complete protection.
No.
But obviously there are other NLRPs out there. Is the cleavage site of DPP9 conserved? And do you think the other NLRPs would contribute to the phenotype of the DPP9 mutants?
Yeah, so far, NLRP1 is the only inflammasome in mice that has been said to be regulated by DPP9. There's a inflammasome also present in humans called CARD8, which is also regulated by DPP9. This is not expressed in mice, so it's not contributing to the mice disease or mouse disease. However, it definitely could be contributing to the human disease. As per the mice, as I did the other inflammasome knockout, so the AS knockout, the GAS-MND knockout, they still only had that incomplete penetrance of rescue, very similar to the NLRP1 knockouts. So from that, I don't think that there are any other inflammasomes that are contributing to the disease in the mice, because if there were, you could imagine that those other inflammasome component knockouts would provide a greater rescue than just the NLRP1 knockouts.
Is the proline of that proline cleavage site conserved though in their sequences?
So I haven't looked to see if that proline motif is present in any other NLRP or any other inflammasome proteins. Yeah.
yep, one more question over there.
Great talk, Cass. I was just wondering, in your DPP9 and then all the different saved mice, was there any residual inflammation?
Yeah. So funny you asked that. We tried really hard to use the rescued IL-1 receptor knockouts. So look at this. So with the thinking that the rescued DPP9 homozygous mutant IL-1 receptor knockouts, they would still have, a fully functional NLRP1 inflammasome pathway. They still should have cytokine secretion. So I tried collecting a bunch of those pups at about 2 days of age. Out of 180-ish pups, I got 2 that were the right genotype. One of the pups had really increased cytokines in their serum. IL-1 beta was increased, as well as a bunch of other pro-inflammatory cytokines. The other pup did not have any increase in any cytokines. I don't know why there was a difference between the two, and because of how incomplete the rescue is, we decided that because we'd already established that the inflammasome is clearly playing an important role, we didn't want to use the mouse lives and resources to collect, another several hundred mice to get the appropriate N numbers.
Yeah, I was just wondering whether you thought that if there was sort of residual inflammation, whether that might be contributing to the runting that you.
You mean residual inflammation in the NLRP1 knockouts?
Yeah.
I did not see any. No.
All right, any last questions? I don't think there's anything on Slido, and if not, all that remains to do is to thank Cass for the completion of her wonderful PhD.